a second. Okay, cool. Can you hear me?
Yeah.
Okay, I can't hear you. Let's see. Can you try speaking of it?
Yeah. Hello.
Oh, there we go. Can you? Got Fabio from Chile. Let me share my screen. You guys see the screen?
Yep.
Awesome. Okay.
Hi, everyone. We'll just give it about 2 minutes for a couple more people to trickle in, and then we'll get started. In the meantime, something fun in the chat box. If y'all want to write where you're attending from, location-wise, that would be interesting. Yeah.
Norman, is the chat turned on? It says chat is turned off for this meeting.
Oh, apologies. All right. I'm gonna start recording. I think it is recording actually. Okay. Thank you, Elsa. All right. Well, welcome everyone to this month's Phantom High-Speed webinar about revealing hidden dynamics with high-speed imaging in microfluidics. I'm Noreen. I'm part of the marketing team, and I'll be monitoring the webinar. Throughout the presentation, if you have any questions, please put them in the chat box, and we will answer them during the Q&A section at the end of the webinar. And so hosting this session is Taka. He recently joined Phantom's Field Applications Engineering team. He defended his PhD from Johns Hopkins University this past February, and his specialty is in microfluidics. So take it away, Taka.
Yeah, thanks for the introduction, Noreen. So you guys can hear me, right? Good. Awesome. Okay, so as Noreen introduced, my name is Taka. I'm a field application engineer here at Phantom. So today I'll be presenting on how high-speed imaging can be used to reveal hidden dynamics in microfluidics. But also how can it be used to reveal hidden dynamics, and also what are the specific techniques and rules of thumb that I typically resort to fully leverage its capabilities. So I'll be referencing two works from my PhD. One looked at how biological cells behave in a microfluidic device, and another one looked at how these alphabet-shaped particles behave similarly in a device. So just to foreshadow a little bit, so assuming this video plays. Awesome. Okay, so one of the phenomena that we observed, for example, is that particles will typically rotate in two orientations. For example, one orientation was where you see the particle alphabet, and another one is perfectly 90-degree offset from that. So high-speed imaging was used to reveal sort of the mechanism and also how particles behave in a channel. So this talk, I'll be talking in context of microfluidics. However, most of the things I mentioned will be applicable to any other microscale system, whether that be a biological cell or purely fluid mechanics phenomena, electronics, you name it. As long as if it's high magnification, high speed imaging, many of the concepts would apply to your specific application. So I want to first start off by introducing what are microfluidic devices. So microfluidic devices are these credit card-sized fluidic devices capable of running different biological assays within it. Because they're so small, there are several advantages that they pose, including the fact that they don't require a lot of material, or they can run assays very quickly. This chip in particular was developed by NASA to test for biological contamination in outer space. And as you can imagine, in space exploration, there are different advantages of having this small form factor, including the fact that you don't have to take that much cargo out there. Now, microfluidic devices can also be used in many clinical applications, and this is more so my specialty. In my PhD, I focused on studying a subcategory of microfluidic devices called inertial microfluidic devices. So inertial microfluidic devices take advantage of a phenomenon called inertial focusing. So inertial focusing is a particle migration phenomenon that occurs when you inject microparticles into a microfluidic device. So these particles, which initially start off in a random position, will migrate to particular streamline locations, and this is purely due to the fluid mechanic forces. So inertial microfluidic devices take advantage of this or some aspect of this phenomenon in order to design for some kind of clinical application. For example, one of the characteristics of inertial focusing is that the distance of the particle from the closest channel wall. So in this schematic, the distance from this left wall to this particle, or the distance from this right wall to this further particle. So this distance will be unique to certain mechanical properties of the particle. For example, what you can see here in the top right here is that softer particles will focus further away compared to rigid particles, or larger particles will focus further away compared to smaller particles. And what you can do is you can design what's called an expansion region that leverages this phenomenon. So what happens in this device in particular is that cancer cells and blood cells are focused in the straight channel, which again is this schematic in the top. The cells focus in a different lateral position, different distance from the wall based off of the mechanical properties. And this expansion region guides the cells to a different outlet based off of the mechanical properties. So effectively, you're able to do a cell sorting in this device. And this is very useful for clinical applications because the alternate modes of sorting cells are typically require some kind of chemical label. However, it requires some kind of chemical label. So, however, in the case of inertial microfluidics, you can do this sorting without any chemical label. You're doing it purely due to the fluid mechanics of the inertial focusing. So then, in order to design any inertial microfluidic devices, you must have some understanding of some aspect of inertial focusing. So that's why in many cases to design these state-of-the-art devices, what you do is you run a scientific study to study some aspect of inertial focusing. And there are different approaches to running these scientific studies. One of the most common approaches is to acquire an image at the outlet of the device. And this is where you get a technical challenge that you must overcome. And the main technical challenge is the fact that the particles are moving relatively quickly. So these particles move at about 1 meter per second. For those of you guys in the audience who work with more macro scale systems like organisms or rockets, 1 meter per second probably seems very slow. However, in microfluidic devices, the window that you image is only about 1 millimeter wide. So because of this, you actually only, if you have an object that's moving at 1 meter per second, all you have is really a millisecond to actually see that particle. And this is a millisecond where the particle is in the field of view. If you wanted to capture 10 frames of the particle moving by, for example, you would have to image at a tenth of this time. And this is where it becomes very useful to have high-speed imaging in order to better understand inertial focusing. And that understanding will drive designing of different applications downstream. Now, having said that, there are actually two alternate techniques that are more commonly used to understand inertial microfluidic system, inertial focusing. One technique is called fluorescent microscopy. So what you do in fluorescent microscopy is that you label your particles with some kind of fluorescent dye, and you image the particles as they flow by. For example, this study conducted by Kim in 2016, they looked at microfluidic devices with a triangular cross-section. They took an image from the bottom of the channel, and they got this fluorescent image in the top right here. So fluorescent microscopy gives you a band where particles generally will focus. It gives you the average behavior of the particles. What you can see here in the bottom right image, which is a high-speed image, is that what's really happening in this device in particular is that you have two focusing positions. So one of the limitations of fluorescent microscopy is that you're going to miss certain nuances that can only be captured in high-speed imaging. And similarly, one of the limitations is the fact that you're going to miss rare events. For example, if 99% of the particles, let's say, focus in this band, and let's say you had a scenario where 1% focus offstream, fluorescent microscopy wouldn't be able to capture this just because it focuses or it images the average behavior of all your particles. So then another parameter that's of interest for inertial focusing is the cross-sectional behavior of the particle. Because right now, this image only covers a 2D snapshot of where the particles are focused. Oftentimes, you want to understand the three-dimensional cross-sectional view of the particle. So one approach to doing this is to image from the side of your channel, and this gives you a similar fluorescent image as shown here. But again, because in fluorescent microscopy, you're getting average behavior, you're missing certain nuances as to, for example, why is this band thicker? Or why is this top band in this triangular channel thicker? So then the workaround to this is an alternate imaging technique called confocal microscopy. It's a type of fluorescent microscopy, but it's more of a laser scanning based technique. And what this allows you to do is allows you to get the cross-sectional understanding of where the particles are located. And now we can see more nuances of the fact that the top focusing position in this triangular channel is actually broken up into two positions. You can view it in the high-speed image, but it's easier to visualize in the confocal image. So in this talk, I'll be comparing these techniques to high-speed imaging and giving you guys some of the advantages and how high-speed imaging can be used to better understand different mechanisms in inertial microfluidics. So in this talk, as I mentioned, I'll be referencing two works from my PhD. One work published in Physics of Fluids in 2023 looked at how biological cells behave in a microfluidic channel with a rectangular cross-section. And the second paper, which is an archive paper, looked at how these alphabet-shaped particles behave again in a microfluidic channel with a rectangular cross-section. So then before continuing, I want to add two disclaimers here. First of all, this work was conducted entirely at Johns Hopkins University under Professor Claire He. So all credit to them. This was done before I joined Phantom. All the information I'm presenting on today is available on the papers published online. So if you guys are interested in more details, you guys are welcome to look them up or shoot us an email. The other thing is that this second paper in the top right is a preprint. So I'm going to be presenting on the version that is available as of February 2026. There may be different versions that come out over the next couple of months. So then going back to the talk, I want to present on these individual studies, give you guys a brief background before I talk about how high-speed imaging was used to better understand the focusing phenomena. To start off with this study that looked at biological cells. So here again, we're looking at how biological cells inertially focus inside a microfluidic channel with a rectangular cross-section. So in this study, we were looking at the microfluidic channel from two perspectives. So we took videos from two different orthogonal views. It's sort of similar to Kim's study, except in our case, we're using high-speed imaging. And what we're trying to ask is what happens to the cross-sectional focusing position of the particle as you change the fluid that you suspend the particles in. And then the second study that I'll be referencing is this study, the archive paper that looked at these alphabet-shaped particles. And here, instead of, we were looking at a microfluidic channel with a rectangular cross-section, but instead here, we're only looking at the particles from one perspective. And the main goal here is to look at rotation because the rotation behavior of non-spherical particles can be quite unique. The motivation behind looking at these different alphabet shapes come from the fact that the current literature looking at non-spherical particles focus almost entirely on these axisymmetric particles, these particles that have axis of symmetry. However, we wanted to look at more complex shapes because many clinically relevant particles, such as microplastics, actually will be more complex in geometry. So this alphabet shape was our attempt to break the axisymmetry and try to understand what happens as you make your particles more complex in their geometries. So in both of these studies, we used high-speed imaging to better understand the actual mechanism behind how particles behave in certain aspects of inertial focusing. Now, this webinar will be broken up into two parts. So in the first part, I'll be talking about how the advantages of using high-speed imaging to reveal hidden dynamics. Basically, when do you want to use high-speed imaging for your own experimental system? And then the second part of this talk, I'll be presenting on specific techniques that I use, general rules of thumbs that I used when I design my experimental system, whether that be on the equipment side or on the actual image processing side. So then starting off with the first part, I'll be presenting on what are the advantages of using high-speed imaging. So basically here, I'll be comparing high-speed imaging in context of fluorescent and confocal imaging. And basically, there are two main advantages of using high-speed imaging. The number one advantage is the fact that you're collecting more parameters, you have a higher dimension of information, and this gives you a powerful tool for gaining deeper insight into mechanism, not only what is happening, but also why certain things happen. For example, if we go to the study looking at the alphabet-shaped particle, one of the parameters that you can collect with high-speed imaging is the orientation of the particle. What is the actual angle the particle is at? This information is not available in fluorescent or confocal microscopy because in the case of fluorescent and confocal microscopy, you're getting average behavior. However, in high-speed imaging, you're getting individual frames, so you're able to get the orientation of the particle as it translates through your device. Now, what's important to notice is that this parameter is very powerful if you combine it with other metrics that you can measure from your videos. For example, in this case of the alphabet study, I mentioned at the beginning that the particles assume two orientations of rotation. One is where you see the cross-sectional alphabet of the particle, and the second one is perfectly 90-degree offset. Now, if you look at the feature size, what you can see is that there are common features among the two orientations, but there are also certain orientations that are unique to each rotation orientation. So then in this study, we were looking at the videos only from one perspective. But what you can do is you can combine the feature size information we get from two orientations to get a 3D rendering of your particle, to calculate how does the particle appear from the wall, or how does the particle appear from the drag area. So you don't actually directly observe these two different perspectives. However, you can do a calculation or a simulation to see what does the drag area look like and what does the wall view of the particle look like. And this is very important because not only now do you see the actual rotation rate of the particle, what is happening to the particle, but you also get an idea of why certain things happen. The drag area gives you some insight into certain mechanisms behind the rotation rate that you observe. So one of the key advantages of high-speed imaging is that you get more parameters that you can measure. And by combining the parameters that you measure, you can run a calculation or a simulation like this that gives you not only what happens, but also why do things happen. So that was the first advantage. The more parameters you collect, the more insight and more deeper insight you get into the mechanism of inertial focusing or any phenomena that you want to observe. The second main advantage of high-speed imaging is that these parameters that you collect can also serve as a feedback loop into your system to design more robust and precise experiments. So then going to my alphabet study again, one of the metrics that you can observe in high-speed imaging is the velocity of the particle. So the distance the particle moves between two frames multiplied by the frame rate will be equal to the flow velocity of that particle. So in our alphabet study, we measured the flow velocity of the particle. Here it's plotted as Reynolds number. It's a dimensionless number. For this case, you can consider it to be flow velocity. Now, what's interesting about this case is that in this study, we actually had a syringe pump that injected the particles. And this syringe pump was actually set at only 4 different flow velocities. In other words, you only had 4 different videos that we collected this speed information from. And yet what you see here is you get a more continuous range of the flow velocities of the particles. So what's occurring here is that when you inject the particles into a microfluidic device, the device, which is composed of a material called PDMS, it's a flexible material. What happens is that when the particles are initially injected, the PDMS flexes, almost serving as a capacitor for fluidic pressure. What happens is there is a steady ramp-up time until particles reach a steady-state flow velocity. So this phenomenon is only observable if you're using high-speed imaging because alternate techniques don't give you the flow velocity of the particle. But in our case of looking at the alphabet-shaped particles, we took advantage of this phenomenon where instead of waiting for the particles to reach a steady state, what we did is we started taking a video immediately after injecting the particles. And this allowed us to get a more continuous range of flow velocities of the particle while only having to take 4 different videos. Now, this characteristics of there being a ramp-up time for particles to reach a steady state, it was an advantage for this alphabet study, but it actually can be a disadvantage if you're trying to design different devices. For example, if you're trying to design a device like the one shown here, where again, particles focus in a different distance from the channel wall, and what you're trying to do is you're trying to find a condition where you can get 2 particles in a different lateral position, different distance from the wall to design a self-sorting device. If you have a scenario, let's say, where you're trying to sort 2 particles, particle type A and type B, shown in a black and purple circle here, if you're using fluorescent microscopy instead of high-speed imaging, for example, you might run an experiment to get that particle type A gives you a band at the center of the channel, and particle type B gets 2 bands at the side of the channel. So then you might conclude this indicates that you can design a self-sorting device because particles are focused in a different lateral position. But the reality is, because you are not measuring flow velocity, you do not know if the particles are focused in a different position because they are different in mechanical properties, or if you just simply did not wait long enough for particles to reach a steady state flow velocity. So this is why it's important to use high-speed imaging, because using high-speed imaging gives you velocity as a measured parameter. And by measuring velocity, you can ensure that your particles are controlled for flow velocity. So then one question you might ask is, okay, then could we just wait an extended amount of time before running your fluorescent images or collecting fluorescent images? That way you could, in principle, use fluorescent microscopy. However, the thing is, you actually cannot measure, you cannot calculate how long you have to wait until particles reach a steady state flow velocity. How long you have to wait for particles to reach a steady state will depend on the exact dimensions of your device, the PDMS material properties, the flow rates, the particle type that you use, the solution that you use, and many other experimental parameters that you simply cannot account for using some formula. So that's why you typically need to run the experiment to see how long it takes for a particle to reach a steady state. And only then can you confidently say you have to wait this amount of time before particles reach a steady state. And that's why it's advantageous often to just use high-speed imaging, so you have that feedback in order to design a more robust and precise experiment. So the two advantages of a high-speed imaging was the fact that you get more parameters, which allow you to get deeper insight into the mechanism behind different phenomena that you observe. And also the second advantage is that parameter can also serve as a feedback loop to design more controlled or precise experiments. Now, of course, these two advantages also come with a trade-off, namely the fact that you're collecting so much information that you need a very more robust architecture to analyze the data. For fluorescent microscopy or confocal microscopy, all you get is one image, so it's relatively low effort to both collect and analyze this data. However, for high-speed imaging, you're collecting sometimes even a terabyte of data per experiment, so it takes a little bit more effort and time to analyze this. I will talk a little bit more about how to overcome these trade-offs in the second part of this talk, webinar. However, here I want to talk a little bit more about the visualization challenges associated with high-speed images, or the data you collect from high-speed images. So here I want to reference the study looking at biological cells. Again, here, instead of taking a video from one perspective, we took videos from two perspectives on our device. And this is an example of one of the plots we got from one of the experimental conditions. And you can immediately see this is a very dense amount of information that you're collecting. So to quickly go through this, the y-axis here is the lateral position. That's the distance of the particle from the closest channel wall. The x-axis is flow velocity, either plotted as the actual flow velocity of the particle or the Reynolds number. The color scheme here is the size of the particle. The plot on the bottom has two color schemes because we took images at two different focal planes. And these individual dots are individual particles that flow by, individual instances. And you can see here, this is quite a lot of information to try to process. I mean, I ran these experiments and I spent hours analyzing this data. And even for me, this is a lot of information to try to digest. And what you have to keep in mind is that this is only one condition out of the 4 conditions in this particular study. So in this study, we had 4 of these dense plots that we had to plot and also communicate to an audience. And on top of this is for biological cells, which are spherical. If you're looking at the alphabet study, you also have rotation rate that you have to look at, as well as feature size and many other dimensions that you would potentially plot on a dense plot such as shown here. So then you can see there's quite a bit of challenge to try to communicate this data to an audience in a set amount of time, whether that be in an academic journal or a conference talk. So then the solution to communicating this kind of dense information is to be a little bit more artistic in a summary figure. For example, those 4 plots that I just showed can be summarized into this one simplified figure. The schematic on the top particularly is important because you see on the axis here that goes from elastic to elastoinertial, and the y-axis here that goes from low elasticity to high elasticity. Basically, these 2 axes represents the four different experimental conditions that we tested for, and the schematic shows key transition points that we observed among these different axes. So by using this schematic, it helps to simplify how long it takes to understand key points in your data. And for example, if I'm at an academic conference or any conference, I will typically present just enough data to show how robust or how much effort we placed into try to thoroughly investigate the inertial focusing phenomena. But in terms of communicating my results, all I typically use is this schematic up here. It contains all the information I need to communicate my data in a set amount of time. So when you collect high-speed imaging data, what you need to do is you need to summarize your data in some kind of simplified schematic, and it typically helps to be a little bit more artistic in how you do that. The other rule of thumb that I like to use, I like to talk to some of my colleagues who aren't directly involved in my project, because that way I get a perspective from an expert who's not necessarily spent hours with my data to get a fresh look. And that fresh look helps you identify what are the points inside your data that are not as clear, and what are the points that don't need extra clarification. So to summarize this first part of the webinar, the 2 advantages of high-speed imaging is that you are able to see more deeper into the actual mechanism behind certain phenomena that you observe. And also, the information that you gather can be used as a feedback loop to better improve the accuracy and also the robustness of your experimental design. The trade-off is that you must place a little bit more effort in both collecting, analyzing, and communicating your results. And particularly, I mentioned that with communicating your results, you must be a little bit more artistic when communicating the key points that you want to communicate. Now, for those of you guys who want the mini cheat sheets to use to decide whether or not high-speed imaging is for you, this is sort of a summary slide that I typically use in order to better understand when is it appropriate to use high-speed imaging and when is it appropriate to stick to more simple techniques. So the first 3 bullet points here at the top are just generally the cases when you just have to use high-speed imaging. Particularly, for example, in my studies, if you're trying to look at rotation, high-speed imaging is really the only way you can look at high-speed imaging of these particles. And therefore, in that case, it's a no-brainer to go to high-speed imaging. However, there are certain cases where it's not as clear whether or not high-speed imaging is appropriate or not. And those occur in the bottom 3 bullet points. For example, if I'm getting an experiment that is not as consistent, for example, if I run an experiment yesterday, I run the same experiment today, and if I'm getting different results, then I would consider using high-speed imaging, although I would also consider just testing for different controls without the high-speed imaging just because of the amount of effort it takes. Also, in many cases, if my experiments are failing, I'll use high-speed imaging as a diagnostic tool to troubleshoot some issues. But those are the cases when I'll be on the edge of whether or not I want to use high-speed imaging or not. And you guys are welcome to ask us questions about whether or not your specific case would require high-speed imaging. And I'd recommend also talking to any of your colleagues. If any of your colleagues use high-speed imaging, I'd recommend asking them for their opinion because using high-speed imaging is quite a bit of commitment. So whether or not it's appropriate is something you want to answer before you actually commit to it. So that was the first part of this talk or this webinar. I presented on how high-speed imaging can be used to reveal sort of hidden dynamics that are typically not obvious. So then in the second part of this talk, I'll present on specific techniques or general frameworks that I use in order to better understand, better leverage the capabilities of these cameras. So in this second part, I'll be presenting on these 5 bullet points. First of all, I'll be presenting on how do you select the right equipment that's appropriate for your setup. Then I'll be talking about how do you actually use that equipment properly. What are the key knobs that you want to be turning on in your equipment? Then I'll be talking about once you acquire the images, what is the general rules of thumb I use when I try to design an image processing workflow to quantify key metrics inside my images. So to start off, I'll be presenting on how do you select the right equipment and also how do you actually use your equipment to get clean and crisp images. There are basically 3 categories of equipment that you need to purchase or you need to at least consider. One is specific to your domain, so that's whatever experimental system that you're trying to analyze. If it's a biological system, this might be a 96-volt plate, or if it's a microfluidic device, it would be your fluidic pressure source or syringe pump and your actual device. The second 2 classes is your optical system. This gives you the high magnification and your camera system. Now, the optical system can either be a microscope. It was a microscope in my PhD, but it can also be just a physical lens you attach to the camera. For the camera system, it's typically advisable to have some kind of data store system. For example, in my experiments, I collected about 1 terabyte of data per experiment. So I just needed some kind of way to offload my data off my computer just to have more space so I can do analysis. It's typically advisable for this data store system to have some redundancy. For example, have 2 of these data stores in 2 physical locations in case one place gets flooded, for example. And then the last piece of equipment that's often overlooked is to select the right illumination source, to select the right light source. So all microscopes, for example, will come with a built-in light source. However, if you try to take a high-speed image using the built-in light source in a microscope, what you get is an image like this where the image is very dark and your particles appear very blurred and blobby. However, if you select for the right light source, what you get is a more clean and crisp image. And the reason why you need this high power light source is because the high-speed camera, the shutter is open for so little time that the time it has for light to go into the camera is so limited that you just need a stronger source of light or light source. And in my opinion, the most important piece of equipment that you should consider when trying to design a high-speed, high-mag experimental system is the light source. Because as you go to higher magnification, as you go to higher speeds, almost automatically you need a higher light source. And this is typically the most important piece that will make or break whether or not you can even use your equipment to collect good high-speed images. So then, assuming you select the right equipment for whatever setup that you're trying to use, the next step is to learn how to use or how to collect correct or clean images that contain the information that you're trying to acquire. So going back to the example of inertial focusing, this schematic is just Maselli's a schematic. I like using it just because of I like the way it looks. But in my physics of fluids paper, when we imaged using the high-speed camera, we're using what's called an inverted microscope. So we were imaging from the bottom of our device. And when you image from the bottom of the device, what you're actually imaging is actually at what's called the focal plane of imaging. So you have this focal plane, this plane inside your device where particles appear crisp and clean as shown here. If your focal plane, for example, is offset from this, what you get is either a blurred out image or a darker image. And either one is not ideal. So the goal with imaging or using any of your equipment is to image such that the focal plane captures your particles of interest or any subject that you're trying to visualize. Now, in order to do that, you have two knobs that you can turn on your microscope. The first one is the focusing knob, which allows you to adjust the location of the focal plane. From my experience on a Nikon microscope, you can get this within a 1 micron resolution. However, how accurately you can adjust the focal plane will depend largely on the actual magnification system you use, the type of microscope or the type of lens that you're using. The second knob that you can turn is the aperture. So what the aperture does is it closes the amount of light that gets to your subject. And the more close your aperture is in terms of focal plane, what it does is it makes your focal plane deeper, or it's wider in the image here. And by closing the aperture, even if your focal plane is offset from your subject, by having a wider focal plane, what you can do is you have a higher probability of getting your particles. So then at a first glance, you may assume that it's always better to close your aperture as much as possible. However, the reality is in microfluidic devices, a lot of times you have contamination or not contamination, background debris inside your system. For example, in the alphabet study, particularly, we had a lot of background debris because these are manufactured particles. So what you're trying to do with aperture is you're trying to optimize the actual depth of your focal plane. You want to make it just large enough that you have a high probability of getting all your particles in the focal plane, but you don't want to make it so large that you have a lot of background debris that also makes its way into the focal plane of imaging. So you're trying to find that right balance to acquire the most high quality images. So to summarize that first point, in terms of equipment, the most important piece of equipment to select for is your light source. Whether or not you have a strong enough light source will make or break whether or not you can collect clean and crisp images. The second thing is, in terms of how do you use your equipment system, the two key knobs to turn is your focusing knob and the aperture. You control these to adjust the focal plane such that you have the highest probability of capturing all the particles that are of interest. So then once you've acquired your images, the next step is to quantify information from that images. In other words, the next step is to develop an image processing pipeline that can process all the data that you collect. So then for me, the general rule of thumb that I use when trying to process the high-speed imaging data is to start with simpler techniques before I move on to more complex state-of-the-art techniques like neural networks. For example, in my alphabet study, one of the tasks that we wanted to look at is we wanted to find a way to segment out our particles and get the orientation of the particle. Now, at a first glance, this sounds like a neural network problem, more specifically an instance segmentation problem where you segment the particle out and get its orientation. However, the reality is you can actually achieve this task using more simpler techniques. For example, what you can do is you can take an image of your device without your particle, image of your device with the particle. By subtracting these two images, you get this background subtracted particle in the bottom right here. Then using simple thresholding, it's very easy to crop out your particle. And then using an edge detection technique documented very well on MathWorks, you can get a clean and crisp outline of your particle. Once you get an outline of your particle, what you can do is you can use a simple cost function to get the orientation of your particle in reference to some reference frame. So here I'm showing how you can use background subtraction, edge detection, and thresholding to get not only the segment of your particle, but also its orientation. And the reason why I prefer to use simpler techniques before using more state-of-the-art techniques is because these simpler techniques typically will be less computationally intensive. And this is important because you're collecting about 1 terabyte of data per experiment, at least in my case, and therefore I need to be pretty efficient in how I process that data. By using these simpler techniques, in my case, I was able to process 1 terabyte of data within 24 to 48 hours on a pretty standard laptop I purchased on Black Friday a couple of years back. Now, having said that, I'm not saying that you should avoid neural networks or like more fancier techniques at all costs. I'm just saying you should start off with the simpler techniques, and if you can get away with them, stick with them. And you should resort to the neural networks or the fancy techniques. Don't make it the other way around. And this is very important when you're trying to handle this amount of information and you're trying to be efficient with how you process the data. So again, the rule of thumb that I use for developing image processing pipelines is to start simple before going complex. That's probably the most important rule of thumb that I use. Now, once you've processed your data, once you've gotten key metrics, quantified key metrics from your data, you might immediately jump to trying to interpret what's going on in your experimental system. However, before you can start interpreting your data, you must also go through one more step, which is the step of validating your data. This is particularly important because at the micron scale, oftentimes you can't directly observe what's going on. It's not like you can put a ruler next to your subject. For example, in my biological cell study, one of the control experiments we did is we focused the particles in 2 focusing streamlines as shown here. And we purposely imaged 3 videos in 3 different focal planes where 2 of them were offset. And one of the metrics that we measured was the diameter of the particle. And one of the controls we did is we took a measurement of the diameter of particle using a handheld scepter device to validate that our diameter measurements are most accurate when the particles are in the focal plane of imaging. And this type of validation is important to do before you even start interpreting your data, because there's a lot of things that can go wrong in your measurements. So making sure you have robust data before you start interpreting it will save you a lot of time in future experiments that you conduct. And once you run the validation, then you can do all the fancier calculations and interpretations you want to do, including this type of analysis, the calculation that allowed us to calculate the drag area of a particle, and also the actual distance of the particle from the closest wall. So to summarize, in this second part of the talk or the webinar, I went through sort of key steps, so key thought processes that I go through from selecting the equipment all the way to validating my experimental results. These rules of thumbs are just general rules of thumbs that I follow. There are other nuances that are still important in trying to design your experiments, but these are the steps that you definitely want to cover before you go for more try to cover more nuances in designing your experiments. So to summarize this webinar, in the first part, I presented on how high-speed imaging can be used to gain deeper insight behind the mechanism of certain phenomena. And then I also explained how the parameters that you collect can be used as a feedback system to better design more precise experiments. And that level of precision is often necessary when you're trying to test the limits of certain pieces of equipment or certain technologies. In the second part of the talk, I presented on key considerations that I make when going from experimental design or selecting your equipment all the way to processing your data. And these general rules of thumbs are just things I learned over the years when trying different things with microfluidic systems. And with that, I will end this webinar. Thank you so much for your attention, and feel free to shoot any questions you have in the chat, and Noreen will answer them for you, or read them off and I will answer them. Thank you so much for your time.
Right. The first question is from Gabriel. He asks, is this technique implemented in the field right now? And if so, how is it being used?
Yeah, so I think you're referring to high-speed imaging to diagnose or high-speed imaging in microfluidics. And the short answer is yes, it is used. So high-speed imaging in microfluidics is sometimes used as a diagnostic equipment. So the example I showed was a cell sorting device. So in that case, you don't necessarily need to image once you design your device. But in some cases, you image the particles as they flow by, and that whatever information you collect about the particle as they flow by will be the measurements you make. It might tell you the particles in a certain disease state, or it will be used as a diagnostic technique in some cases.
Another question from Pierre, I think. Did you also try high-speed microholography?
I personally did not. I know about holographic imaging. I'm not familiar if there is a technique for high-speed holography. Um, but yeah, I personally have not tried it. It's a very interesting technique, though. For those of you guys who don't know, it's a technique that allows you to get more directly measure three-dimensional information inside a particle that's flowing by without having to take those two videos like I did. So it basically cuts the amount of amount of data that you need in half. Although I think on the processing side, it becomes a little bit more complicated. But I personally have not tried it.
Question from Roy. Does high intensity illumination produce heat that affects the experiment?
Yeah, so it does not produce, it does produce heat, but it's not enough that it would like boil my cells, for example. The particles are flowing by within a millisecond, and even in the field, the area that it's being heated at, the fluid is moving relatively quickly, so there's no detrimental heat effect. If you're running like a test in a 96 well plate, for example, and you're trying to image with this high illumination source for hours, it could eventually result in heating of your cells. But in microfluidics, because you're flowing particles by, it's not a big issue, at least in my applications.
Question from Chen Yi. Can you comment on high-speed images together with fluorescence, especially in terms of average versus non-average behaviors?
Yeah, so I think so. I guess there are two ways this question might be asking for. So you're either asking for if high-speed imaging can be used, like you can do high-speed fluorescent imaging. And then I guess the other thing you might be asking is when would you use both techniques for designing an experiment? So I'll answer the second interpretation first and then the first interpretation. So then fluorescent microscopy is a very low effort measurement system. So basically you can take, all you need is one image. So it takes maybe a minute to analyze your data. So sometimes it's used as a way to scan different experimental conditions before doing high-speed imaging. Having said that, in biological systems, oftentimes you don't really necessarily want to use a fluorescent, you don't necessarily want to label your cell with a fluorescent dye because sometimes that can lead to off-target effects that will sort of bias your results. But fluorescent imaging to get the average behavior is sometimes used to just quickly scan for different experimental conditions before committing to high-speed imaging. And then the second interpretation is that you might be asking whether or not high-speed imaging can be used, if you can do high-speed fluorescent imaging. The short answer is in some scenarios you can, but it's very difficult. So as I mentioned, with high-speed imaging with regular bright field, you need a very strong light source to be able to image the particle flowing by. And the same principle applies to fluorescent imaging. However, to get a stronger fluorescent signal, you only have two options. One is to hit the particle with a stronger laser source, but then that has issues like it'll kill the cell in some cases or lead to photo bleaching. So that's oftentimes not a good option. So then the alternate is to come up with like a state-of-the-art fluorescent label. In that case, you might be able to get enough fluorescent signal to get a high-speed image of the particle, but even then, it's a very hard technique to actually implement in practice. I hope that answered your question.
Question from Daniel. For the blood flow experiment with the 3 channels filtering cancer cells, is it known why the cancer cells flow differently than the non-cancerous cells?
Yes, so in that study in particular, what they showed is that cancer cells are actually larger and they're also softer. So larger particles focus further away from the channel wall, and softer particles also focus further away. So these cancer cells that just happen to be larger and softer, so they're just focused further away from the wall, and that's why they flow to a different outlet. Good. Any other questions? Yes, not.
Well, if there are not any more questions, yes, this video is recorded and it will be sent out right after the end.
Actually, there's another question from Daniel.
Yes.
You can read that. I don't want to take your job, so.
He asked for the L-shaped object flowing on the wall, was the L contacting the wall?
Yeah, no. So in the case of these alphabet studies, they weren't contacting the wall. There were some cases where it sort of appeared to contact the wall, but that was more so because there's like another particle in the field of view and like it dislodged it from its spin line to make it hit the wall. In that study in particular, we were looking at particles without, like, we weren't looking at particles interacting with other particles. We wanted to see individual particles. So that data was exempt from the analysis in that case, because it's just a completely different phenomenon if you're talking about particle-particle interaction. But yeah, in that case, it wasn't interacting, or it wasn't touching the wall per se. It got very close, but it wasn't necessarily touching it.
Question from Roy, how were the alphabet particles created?
Yeah, it was created using what's called stop flow lithography. It's created actually by a collaborator over in South Korea. So basically the technology is that you have a mask of these alphabet shapes, and then light passes through only through that alphabet shape location. And then what you do is you get this light sensitive polymer that flows by. You shine light, which polymerizes the particle to create the particle. It flows by, and you just repeat that over and over until you get 100 or whatever number of particles that you want to develop. And it's inside the paper. There should be a reference. One of the references will cite our collaborators' work that sort of talks in detail about how to actually manufacture these particles. Any other last minute questions? Yeah, I guess Daniel, it does look like it walked a little bit, but then in general, it's very hard for particles to touch the wall during flow just because of how strong the fluid forces come from like as you get closer to the wall, you have what's called the wall lift force. So it just becomes very hard for particles to actually physically touch the wall in many scenarios.
Right? If there are no more questions, then I think we can wrap up the webinar. So thank you everyone for attending. And the recording of the live session will be sent out right after if you would like to look at it. So yeah, I hope everybody has a great rest of your day. Great job, Taka. That was a very interesting seminar. So many things that I didn't even know about. But yeah, so we'll see everybody soon.
Yeah,
Later.
thanks for everyone's time.